dignitaries, distinguished guests, colleagues, project outsourcing staff, administrative staffs, and students of ICMR and NIPCR. It is a moment of immense pride and privilege to welcome you all to the 4th Foundation Day celebrations of ICMR National Institute for Preclinical Research.
శుభోదయం అందరికీ. సో మన 4వ ఫౌండేషన్ డే NIPCR సెలబ్రేషన్స్కి విచ్చేసిన అతిథులకి ఆహ్వానం.
To commence this prestigious occasion, I welcome all the dignitaries over the dais. It's my proud privilege to invite our chief guest, Dr. Kumaraswamy Thangaraj, Padma Shri awardee, an eminent scientist whose pioneering contributions in the field of genetics and molecular biology have significantly advanced our understanding of human health and disease. Please, sir, book it. It is indeed an honor to invite our guest of honor, Dr. Naveen Kumar, whose valuable contributions and associations with scientific community contribute to inspire excellence and innovation. And now I invite our director, sir, ICMR and NIPCR, Dr. Mukesh, sir, whose visionary leadership has guided us always. So now we all will stand for a Vande Mataram. Today we are here not merely commemorating a date, we are celebrating a journey of scientific curiosity, ethical research, and translational excellence that contributes directly to the nation's healthcare system. At NIPCR, every experiment, every dataset, and every innovation reflects the spirit where rigorous scientific methodology meets the larger vision of national health advancement. With the spirit of knowledge, innovation, and service, let us formally commence the celebrations of this significant day with the ceremonial lighting of the lamp. I request all the dignitaries on the dais to join for the lamp lighting ceremony, and P. Suresh sir also for the lamp lighting. ज्ञान का प्रकाश होता है, वहाँ अज्ञान का अंधकार शब्द ही समाप्त हो जाता है। Now I request our director, sir, for a welcome address. Sir, please.
Distinguished guests, the Director General of and Secretary Director General ICMR and the Secretary DHR shortly joining. Our chief guest, Dr. Kumar Swamy Tangaraj, the Padma Shri awardee and very renowned scientist of this country, to which we all are proud. Our own Director of NIV, National Institute of Virology, Dr. Naveen Kumar. My colleagues who have joined online, different directors. My colleagues who are sitting in the dais, our founding director in charge, Sri Dr. Suresh Pothani ji. Online just now we joined with our past director, immediate past director, Dr. S.G. Ramachandra, Dr. Bharti Kulkarni, the director of NIN Hyderabad, the institute from where we have actually originated, our mother institute. My fellow scientists, my admin staffs, AOs, ACOs, senior AO, my support staffs, my dear students, my technical staffs, and all other fellows who are present here, ladies and gentlemen, and the students who are here. A very good morning to all of you, and congratulations to all of us, including myself, that we are standing today on the 4th foundation day of this institute, to which I think we can take all pride, and soon we will become the national pride in becoming the National Institute for Preclinical Research, which is the largest preclinical institute. And we follow the highest level of ethical standards that any institute can follow in the field of preclinical research. As we all know, we were inaugurated in December 2022, and now we are in 2026, 4 years down the line. And in these 4 years, we have taken leaps and bounds to become nearly fully functional. And we range entire range of laboratory animals than one may require in current or in future for any clinical trial for the drugs, vaccines, medical devices, biologicals, and any other things which is of public importance. We also included in our range of animals that might be required for our preparedness towards the future pandemics such as COVID-19 that we have. I'm very glad to share with all of us in these 4 years, while we expanded our infrastructure, we have also started working vigorously in research. Our scientists, we are just 10 scientists, 2 more has added this year. And just in last 1 year, our scientists have published 20 SCI index journals, some of which are impact factor as high as 8.5. We have publications in journals like Archives of Microbiology, Molecular Biology Reports, IJBM, Journal of Medical Microbiology, Journal of Medical Mycology, and so on. We have also, our works have been presented in last year itself, 7 international conferences, some of which includes the BioHeal organized by the IIT Roorkee, the Virocon organized by the NIV Pune and Indian Virological Society, and the Congress organized by the Indian Veterinary Council. Our scientists in last 2 years have also participated and secured 1 patent along with the JIPMER Pondicherry, and we have filed 1 more patent along with the NIRT Chennai. So this is a very good achievement on part of our institution in just last 1 year. And I'm also very glad to share that we have currently 28 research projects which are ongoing currently with the worth of 5 crore for this year alone. And it includes, it excludes the 3 ANRF projects that we have been sanctioned and 1 DBT projects that have been sanctioned to our scientists. So I take proud in that. We work in a wide range of areas like viral research of vaccines. Along with NIB Pune, we have been developing the animal models for the Chandipura virus, for example, and trying to test the drugs and vaccines. Working on mRNA-based vaccine evaluation for dengue. We are working on the host pathogen interaction for the Cassino forest disease virus. We are also working on developing animal models. We have acquired the bonnets, which are the good models for the KFD disease. We have established the facility for the ferrets, and this year we hope that we'll have the ferrets, the only facility for the entire country to have the ferrets for work on influenza research. We also have established the, working on establishing the models, for genetically engineered models for measles, rare genetic diseases such as Suzanne diseases, Parkinson diseases, and we have established the system for the organoid development. And I'm very proud to say that our institute is capable of providing iPSC derived organoid for almost every mouse organs that one may need. We also work on the non-communicable metabolic diseases, such as we are developing the goat models for osteoporosis, human osteoporosis, diet-induced MSLD, which is a liver disease in rhesus macaque. We are developing pig atherosclerosis model and trying to evaluate the impact of stress and smoking on developing the cardiovascular diseases. We are developing models for the ulcerative colitis in pigs. We also work on reducing our cost burden on developing the indigenous diagnostic kits, screening kits for multiplexing, multiplex detection of the range of the pathogens that we do. In all our animals that we have is lamb-grown, purpose-bred SPF animals, in which we between 20 to 25 pathogens are screened every 3 months by sentinel monitoring. We incur a lot of cost for that, and we are working on developing our own indigenous kits to reduce our dependence on the import. We also work with other institutions. We also support to other institutions, like we have provided our support to IIT Delhi for developing a multi-material screw for orthopedics. We have done the POC validation of jugular venous pressure-guided diuretic device in goats for the cardiac care center in Bikaner. We have developed the first time in the country the hyperbilirubemic pig model and evaluated a nanoformulation which can be used for treatment of neonatal hyperbilirubinemia in humans with the help of ICMR. Apart from that, we are also doing research for other institutions like CDRI. We are working with companies like the Maxwell Biotech. We are working with NDDB, IAL. We are working with the Bharat Biotech. We are working, and there are a few more companies. We have generated, within this year itself, we have generated a revenue of approximately 2 crores from the sale of animals and the other services that we provide to our collaborators. This is in addition to collaborative research that we do, like we have the 35 lakhs projects going on for evaluating the colorectal carcinoma, cytotherapy, CAR-T therapy we are evaluating. We have 3 projects ongoing on evaluation of CAR-T therapy. And I'm very proud to say that currently our institution is the only institution in the country who is capable of evaluating the cell therapies such as CAR-T and CAR-NK. There is no other institution, to best of my knowledge, who can do this evaluation. We also do collaboration with other institutions. In last few years, since its inception, we have also built up our infrastructure. We have, for example, developed our surgical excellence in developing state-of-the-art surgical suite in the pig and NHPs. our capability can be gauged by the you will be glad to know that next month hopefully we'll be doing the LVAD transplantation, the left ventricular assisted device or artificial heart as we say it in our sheep model in our country. We also have set up new laboratories like immunohistochemistry laboratories. We have commissioned the molecular virology and serological laboratory, specialized organoid laboratory. We have introduced advanced in vitro animal imaging systems, including the microCT, which is coming up shortly. We have installed the confocal microscope, GCMS, and few other equipment that is in the pipeline. During this time, we have also acquired the accreditation for the good laboratory practices from the NGCMA for immunogenicity and subchronic toxicity studies in pig, and we are also progressing towards the dogs and NHP study, which is a certification that what we are doing is of internationally accepted data sets. And we have also, I'm glad to announce or inform the others, those who are not aware, our scientists have also brought laurels to institute. Two of our scientists, Dr. Varinder Singh Raina and Dr. Gunaji Balakrishna Kumbhar, have received the Young Scientist Award from one of the conference in IIT Roorkee. And Dr. Gunaji Kumbhar also received Gau Gyan Paritoshik Award at IVRI, Indian Veterinary Research Institute, from the Indian Association for Advancement of Veterinary Research. Besides this, we have also been engaging with the other professional bodies. We have been engaging with the communities. Tomorrow we have program for the NABL accreditation awareness program for the institution in Hyderabad. We also, under the flagship program of ICMR called SIGN, we have created 10 mini labs where we brought the students from the other schools and gave them the live experience how they feel one day as a scientist. That was very, very appreciated by schools like DPS. And we are receiving now multiple requests for them. We keep requests and companies keep visiting from nearby companies, pharmacy colleges, veterinary colleges. They keep visiting and we open our institute for them. We also work on the capacity buildings for training on biosafety, GLP principles, OECD principle, mainly for our internal staffs, but also for the external staffs as well. We are also the key knowledge partner for the MedTech Mitra, which is a flagship joint initiative of the ICMR, along with the Niti Aayog, CEDESCO, the GEM, the Department of Pharmaceutics, and few other organizations. During that, we provide assistance in understanding the preclinical requirements for various innovations, drugs, medical devices, mainly focus on the medical devices, but we also provide assistance for the biologicals, the monoclonal antibodies, for the drugs sometimes, and for the LVAD devices. And I'm very proud that we'll be expanding this to further by our own setup for how we can take this matter, this assistance, to the researchers of the school, the academic institutions, to researchers of the research institutions, and the young startups who are oblivious to the regulatory requirement. Generally, as we all know, that at the research bench in a university setup or a research setup, when we do the research and few animal studies, we tend to think that we have done. But that's just the beginning of the problem. So we have started our own program to assist them, and our Director General ICMR, when he joins shortly, will be giving more inputs on what we do in that to join. Our institution has taken since then leaps and bounds, and we have, as I was saying, now a resource of 11 different species. All of them are SPF quality animals. We largely keep it for our purpose, for our national preparedness, but we also collaborate with other institutions. We provide it to other institutions for doing the preclinical research on them. And our institutions, besides this, the providing animals, we also, as we said, work with the several companies, several institutions, and I'm very glad to share that these research has been fruitful, and 2 of the products are at advanced level of going into market through the MedTech Mitra program. That is a brief about our institution, and I'm very glad that today we have 2 renowned persons in this field. I may say 3 and 2 joined online 5 persons we have. We have Dr. Tangaraj, and Dr. Tangaraj is, I'll take a brief moment to introduce Dr. Tangaraj. He's a Bhatnagar awardee, which is a very, very prestigious award. But beyond Bhatnagar awardee, he's the recipient of several other awards, which includes the most sought after and most prestigious is the Padma Bhushan, Padma Shri award for this. And he has J.C. Bose Fellowship, Sun Pharma Research Award, Legend of the Hyderabad, which we all know, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Science Award, C.V. Raman Memorial Lecture Award, Dr. U.N. Brahmachari Award, and so on to do long. He's a fellow of all 3 national science academies, which itself marks that one. We have joined with our Director General ICMR and the Secretary of DHR. On behalf of the Institute, we welcome you, sir, and we extend our thanks for taking your precious time. We know that you are very, very busy and you have taken time to join us and shower your wisdom to our scientists. Thank you, sir. It's over to you.
Thank you very much, Dr. Mukesh Gupta. Dr. Mukesh Gupta, the distinguished chief guest of this function, many people who are in the room, and also I can see online Dr. Ramachandra, Dr. Bharti Kulkarni, Dr. Manisha Srivastav, all the colleagues. It is a great pleasure to talk to you today on this day of it's an important day for NARF, which is now National Institute for Preclinical Research, a very important institution for our country now, and something which gives a very different flavor to the Indian Council of Medical Research. Establishment of this particular institution signals a very important shift in the way ICMR functions. Because few years ago, the pre-COVID years, mostly we used to do research, and most of that research was largely descriptive in nature and trying to say what is the problem, what is the extent of the problem, what is the what do we know about the diets, anything which we wanted to, it was largely descriptive. COVID changed that. And post-COVID, we have continued to change that. And this institution was inaugurated in late 2022, 2023, 22, I think, November, December 22, that we, in these 3-4 years, it has come a long way. Now it provides us with the ability to become part of the innovation pathway that will lead to products. And it gives a lot of hope to a lot of academic institutions in the country, whether they are ICMR research institutions or research and academic institutions outside ICM. Because now what we can do is we can help them in that process of preclinical assessment of innovations, whether they are drugs, vaccines, devices. It gives confidence to us of a very different kind. We are already involving the Institute in multiple initiatives. You heard just from Dr. Gupta uh, related to MedTech Mitra, for example, intent, where our goal is to help innovators. This institute is helping NIV with, let's say, new vaccine development, monoclonal antibody development, vaccine for, let's say, Casanova forest disease, antibody for monoclonal antibody for Nipah, etc. So I really see that this is a great institution that can become even greater. It is beautiful. It has been built on a large piece of land. Buildings are absolutely magnificent. The facilities are excellent. Now, they are also, last time I came, I saw a big change. There were also a lot of animals now, and some experimentation is already ongoing. More than 5 studies were ongoing at that time. So that is really good. GLP, we've just heard, we are very happy that the GLP certification has been received for the pig facility. We would like to have it for other facilities, rodents, dogs, non-human primates. And we have to strive to work towards that. I'm also very happy to tell you that finally we also got some scientific and other staff approved by the Department of Expenditure. Previously, most other than the director, almost everybody who was there was on borrowed this thing, because we have borrowed from other jobs in other parts of the ICMR to provide the few scientists. But now we have several posts that have been approved. They are not exactly what we had asked, but still there is several jobs that have been approved. So my request to all the staff, everybody, today is a day of celebration for you all, but also a day of commitment that you should work very hard and in a very collaborative way with multiple institutions. Because collaboration is, if you do not collaborate, your institution will be largely useless, because it is only with those collaborations that you will be useful in this process of making India stronger in research and innovation, particularly in the innovation pipeline. So, with those words, I again warmly welcome all the chief distinguished guests, but also I wish all the staff and the others associated with this institution, including people who were there and have contributed to its development but are no longer there. I wish you all a very happy Foundation Day, and wish that and expect that this institution will continue to achieve greater glory. Thank you. Jai Hind.
Thank you, sir, for your wishes to our institutions, and you have been always a support system for us. Whenever we needed it, you were there. And now, very also proud to announce the launch of the NIPCR QuickAssist by our Director General ICMR. Sir, this special provisions we have made in our institution so that the researchers from the academic institution, the researchers from the research institutions, and many of the startups who are not aware of the preclinical requirements, we made a one-step solution for them. They can visit our website and they can raise their query, and we will resolve them in 24 hours, and if needed we'll provide access to our pool of scientists for discussion. So we request you to, sir, please inaugurate this lunch by making a click.
Thank you. And I think this is a great initiative. I'm very happy to inaugurate and launch it. Thank you.
Thank you very much, sir.
Thank you, sir. This QuickAssist launch marks another step in strengthening the institutional excellence and scientific productivity. We at ICMR NIPCR commit for innovation-driven research support to all others. So now I respectfully invite Dr. Kumar Swami Thangaraj sir, Padma Shri awardee, to deliver the oration of the day.
So, I take the privilege of introducing our chief guest today, who is also my PhD mentor. So, Dr. Thangaraj. So, as already told by our director, sir, he's CSIR Bhatnagar Fellow at CCMB Hyderabad. He was director for CDFD, and he was also holding director additional charge of the Institute for Stem Cell Science and Regenerative Medicine, INSTEM, Bangalore. His main research interest for the last 3 decades has been in the field of population and medical genomics. He has published more than 300 research articles, and he is a fellow of all the academies, as told earlier. He is recipient of several awards, so including the Padma Shri, Rashtriya Vigyan Purushkar, Vigyan Shri, J.C. Bose Fellowship, Sun Pharma Research Award in Medical Science, Legend of Hyderabad, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, Sir C.V. Raman Memorial Lecture Award, and many, many more. So I was fortunate to witness his glory in science while I was in the lab. So he has many more awards. So I warmly welcome him to deliver the oration. Sir, please welcome.
Respected Director General, I hope they're online, Director of NIPCR, Director NIV, a friend Dr. Suresh, all the scientific and non-scientific staff. I hope Dr. Ramachandra and Dr. Bharati also joined online. Very good morning to all of you. And it's my pleasure to be part of this Foundation Day celebrations. And on this occasion, and my best wishes to the director and all of you here for whatever the institute is doing for the last few years of its emergence. I think it has a lot more to offer to the country, every academic and scientific institutes, and also to the pharma companies and um, many more people. So in that sense, wish you all the very best. I think the best to yet to come, because it's a beginning, 4 years, and I think all the staffs are also very young. I have lot more to play for the development of this institute and ultimately to the country. So with these few words, I would like to start my talk. I kept the title as our origin, culture. When talk about culture, there are multiple culture. Every region, if you go, there's a own culture in terms of their habit, food habit, practice of marriage, dress, everything is very unique, right? So I'm going to talk to you about one particular culture, that's about the endogamy, right? Marrying within the community, which is common across India, or you can say South Asia. And what is the consequences of that is the one which I am going to talk to you at the end. When we talk about our own origin, I think you all must have studied, even at the school level, that we have diverged from a common ancestor. We means human, and the chimpanzee from a common ancestor called hominids about 6 to 7 million years. And during the 6 to 7 million years, what had happened? There are several species have evolved, and none of them are existing now, including the Neanderthals, very close relative in terms of evolution. So in this process, there are several changes that happened anatomically. Our ability to walk with 2 legs, ability to make tools for hunting for the food, and also our brain size has increased tremendously. If you look at the early species Chandensis, the brain size was as similar as chimpanzee and has increased to 35% in Australopithecus. Then Homo habilis, it was 50% and gradually has increased. And interesting to know that in Neanderthals, the brain size was slightly larger than human, but we don't know whether they were intelligent than human or not. But this species also know more. Right? But interestingly, several hypotheses have been proposed why this species is no more, how it has vanished. So one of the interesting hypothesis made by Paul Millers from Cambridge, he says that inherent violent behavior of modern human probably responsible for vanishing of this species. I hope all of you are agree that how violent we are and could be responsible for that. And this actually, there's a lot of work in the recent past, particularly Savante Pabo and his colleagues have done in Neanderthal. They sequenced the bones and fossils and established that about 500,000 years back, human and Neanderthal have diverged. They also identified a place where human, Neanderthals, and also Denisovan lived together, and some of the bones which they found have 50% of human, 50% of Neanderthals, and so on. So that gives actually the missing link of human evolution. One would ask question that if we have diverged from Neanderthal, whether we all carry some proportion of Neanderthal genome. Yes, indeed, in one of our studies where of course led by David Reich, and we were part of it, including Seventh Pebo, who has been awarded Nobel Prize in 2022 for his work on particularly Neanderthals, and we established that the human, the modern human who live outside Africa all carry up to 3% of Neanderthal genome. In fact, recently when we sequenced 10,000 human genome, when we started analyzing, a different population has different proportion of Neanderthal genome, suggesting that yes, evolution has took place and we have diverged from Neanderthal about 500,000 years back. So this is to give you some idea about how we have evolved, and if you look at how the modern human evolved, and again establish that the modern human originated in Africa about 160,000 years back. And from Africa, because all evidence suggesting that the bone sample, old bone sample is present only in Africa, there is no other place, although there was some multiple theories proposed, but there was no evidence, scientific evidence to suggest that the modern human was in the early years was found in any other continent. So now one can ask question, from Africa, how did they migrate and populate other place? And there is one hypothesis suggesting that the modern human must have taken the northern land route. Probably there is a connections, they must have walked through the land and populated for the very first time either in Europe or Middle East or something. But there was not much support for the evidence. Therefore, alternate hypothesis has been proposed by archaeologists, anthropologists, historian, linguists, and including geneticists. They say that the first out of Africa human must have taken the southern coastal route, right? All because of some of the evidence suggesting that modern human must have arrived Australia about 50,000 years ago, China 39,000 years ago, Middle East 47,000 years ago, and Europe about 40,000 years ago. But unfortunately, we do not have any archaeological evidence suggests that when did the modern human arrived in Indian subcontinent. There are some evidence suggesting that modern human must have lived in a place like Jolapuram in Andhra Pradesh, but there was no bone samples, only stone tools which predicted that human must have used that. Other than that, we have another evidence of Rakigiri, that is in Harappan region. That sample is also not very old, about 3000 BCE, but it consists of Iranian hunter gatherers, but not Iranian farmers, and anyway again it is not telling anything about the early human migration. So, in the absence of archaeological evidence or ancient DNA samples, we thought of using the contemporary population, means those we all the contemporary population, the present day population. We used different approaches, genetic approaches. One is looking at the mitochondrial DNA. The reason is we all inherit mitochondria and mitochondrial DNA only from the mother. There is no paternal contribution of mitochondria. Therefore, one can trace the maternal lineage by using the mitochondrial DNA. Other one is using the tiny chromosome, Y chromosome, which only male has, because the genes SRY and many other genes which determines the male sex is present only on Y chromosome, and the Y chromosomes are transmitted through male lineage from grandfather to father, father to son, son to grandson. Therefore, by looking at the marker which are existing on the Y chromosome, one can trace the paternal lineage, okay, maternal lineage for mitochondria, Y chromosome for paternal lineage. We also use the other autosomal markers where we can assess the contribution of both the parents, and in combination of all three, we can able to achieve much better results. So, I am going to tell you about very few studies which actually connects our evolution and the disease. Talk about very few selected publications, and from here I am going to take very few examples that how some of these studies has helped us in understanding our own origin. Okay. That is, although we are all not really in human genetics, but we all understand, and curiosity is in many people, those who are not in science, those who are in different fields also is very interested in knowing about our own origin. So, initial study by looking at the tribal populations of Andaman and Nicobar Islands. I do not know how many of you are aware that in India there are typical population groups, they look like Africans. Okay. So, with the help of again one of the ICMR institute in Fort Blair Regional Medical Research Center, we had earlier collaboration, and we went and collected large number of samples. Of course, the sample size is very small, the population size itself. There are 4 tribal groups. One is Great Andamanis, Onge, Sentinelese and Jarvas. These are the 4 tribal groups who typically look like Africans like this. So, we had an opportunity to collect 3 populations. Sentinelese are still in isolation, nobody can go near them, they will shoot with arrow. Okay. That happened multiple times in the very recent past. There are 2 other populations, Nicobaris and Champens. They typically look like Southeast Asian population. Right. So, we analyzed those 2 as well. In this study, initially we looked at only the small proportion of mitochondrial DNA, then we sequence the complete mitochondrial DNA. So, based on the mutations which are present in the mitochondrial DNA, one can able to group them into multiple groups. So, when we sequence the mitochondrial DNA of Andamanis, we would expect that their haplogroup would fall along with the Africans, but unfortunately, we can't say unfortunately or fortunately, to our surprise. So, they were not falling in any of the African specific haplogroups, rather they have acquired large number of mutations. Therefore, we assigned a new haplogroup for them like M31 and M32, and we also calculated the age of the population based on the mutation rate. Right. So, based on that, we proposed that probably the Andamanis are the first modern human who have migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. So, this is the first genetic evidence suggesting that the Andaman tribes are the first modern human who have migrated out of Africa and reached Andaman and Nicobar Islands about 65,000 years back. And this is still holding true, there is no contradiction about this finding. Right. Subsequently, there are many population studies we did, and now I am going to talk to you about another study where we have looked at the 1 million genetic markers across the genome. Right. So, this is disturbing this, I do not know whether. Let me just see. No, not charging. There is a this one, some alert. Okay. So, this was after human genome sequencing was done, 1 million genetic markers were identified across the genome, which are highly polymorphic. So, we use that, and in this study we in fact reconstructed the Indian population history. Okay. So what we suggested was that in the prehistoric India, there were two founding populations. One, we call them as ancestral South Indians, probably they are part of the early human migration when the Andamanis were migrating, taking southern coastal route. One group has stayed in southern part of India, whom we call as ancestral South Indian. There was second wave of migration which took people towards north, and probably somewhere there's a split. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indians. And these two groups later admixed and gave rise to many population groups. So for that I will come a bit later. On the other side, if you look at, these are all Indian population groups. These are European HapMap samples. These are African HapMap samples. These are Chinese HapMap samples. So this gives you idea about the population in India. While we have population, every single population clustering independently, whereas these three HapMap samples, although there are hundreds of individuals, they are in a single cluster, suggesting that genetically those three populations, Europeans, Africans and Chinese, are in a single cluster. That means there is not much difference between the individual genetically, therefore they are in a single cluster. Whereas every single population in India, although forming a single clusters, but there is a kind of affinities between different population groups. This is all because of the admixture. So one good example which I take is taking our skin color, right? That itself will give you some idea about the admixture, how we are admixed. We have extreme dark color, we have extreme lighter skin color, in between there are so many shades, right? So you don't see that kind of skin color in many other countries or continents. Okay, that's the best example which I always give, gives you some idea about how the populations are admixed. There is no single population say that distinctly present except the Andaman tribes. So I am coming back here that I told you that there were two founding groups and these two groups have admixed and gave rise to many population. To understand that, we took another study when this admixture must have took place. As I mentioned initially, there were two founding population, one is ancestral South Indians and the other one is ancestral North Indians. Initially, these two independently gave rise to many population. You can imagine that initial group has settled in a jungle and they have to hunt for food. After completing or finishing half of the food or completely utilizing the food, one group, those who are active, must have migrated to other place, from there to other place. That is how both ancestral South Indian and ancestral North Indians have gave rise to multiple populations. Later stage, during the last 2 to 4,000 years, these two groups have admixed. The color indicates that how proportionately both ancestral South Indian and ancestral North Indians have admixed. After admixing, every single population now in India for the last 2000 years practicing endogamy. Endogamy means marrying within the group. All of you are aware that every single population from whom belongs to maintain very strict endogamy, right? So that is the culture which I am going to talk to you. So last 2000 years of endogamy has left lot of things in the population genetically. To understand that, we took another study where we used 2,800 samples from 275 populations. This time we did not restrict our study with South Indians, we also took sample from South Asia. The reason why we wanted to extend this study to other South Asian countries, or there are some examples from our own study suggesting that some of the disease causing mutations are restricted to only in India and South Asia, but not in other countries. This is one example where we have looked
Based
at
upon the
several
right, that's
cardiomyopathy
fair
patients.
enough.
That's one of the heart disease.
ಏನು
There's a
US ಎಲ್ಲ
25
ಮಾಡ್ಕೊಂಡು
base pair
ಹುಡುಗರು
deletion in a gene called myosin binding protein C3. When we try to screen for that mutation across India, we found 4.5% are carrying this mutation. The mutation exists in a heterozygous condition. It won't cause any disease until 40 or 45 years, but later stage it will cause the disease. But if it exists in a homozygous condition, means both the copy of the chromosome carry the mutation, then the individual die at very early age, right? So by looking at the postmortem tissue of the children who died, we found that both dilated and hypertrophic cardiomyopathy are carrying the mutation in a homozygous condition. Okay. And the reason why I am saying here, try to screen for that mutation across the world, we found the mutation is present only in South Asian countries, but not in other countries. So that's the reason we wanted to take this study to understand the impact of endogamy in South Asian countries. So what we found was, in this is called IBD score, identity by descent. So this graph, if you see, every single population is represented by a single line. That means there are 275 population, 275 lines are there. Some population the IBD score is very long, some population is very tiny. By looking at this, you can actually predict the population history. We can also predict whether the populations are expected to have a recessive disease. If the line is longer, as you see here, that means most of the individuals from the populations are emerged from a single parent. That means most of them have the same haplotype. Okay. When you have very high IBD score, that means, for example, 100 individuals you are analyzing, maybe 90% of them, 90 individual will have the same haplotype. If you take a 10 kb region, there won't be much difference. So that itself suggests that all of them, all the 90 have come from a common ancestor. Therefore, if there is any recessive mutation, it is going to circulate in the population. A recessive mutation in the sense that if the mutation present in a heterozygous condition, that means one copy of the chromosome carry the mutation, other one is normal, the individual will be absolutely fine, there is no problem at all. But the mutation happen to be homozygous, that means two heterozygous individuals marry, there is a 25% of chance that they contribute, both the parent contribute the abnormal chromosome. Therefore, the child has the homozygous mutation will have the disease. So that condition is very high among the people who have high IBD score. But these ones have lesser IBD, that means out of 100, maybe 5 of them must have come from a common ancestor, have the haplotype. Now interestingly, what you see here, two populations which are not Indians or South Asian, one is Ashkenazi Jewish, other one is Finnish population, has been included in this study, but they have tiny IBD score compared to the large populations of South Asia, but still they have large number of diseases. This is Ashkenazi Jewish, this is Finnish population. You can see most of them are autosomal recessive in nature, right? And to give you some additional idea that how it has its impact on causing the disease, look at this pedigree. I hope all of you are aware, circle means female, this is a male. If we put the line, that means these two are married, and put the line down, and if we put square, that means son. If you put circle, is a daughter. So this is heterozygous mutation. One chromosome, one of the pairs carrying the mutation, other one is normal, and he doesn't have the mutation, right? So in this scenario, what happens? They have two daughters, and the abnormal chromosome is going from mother to next generation. Again, it's a heterozygous condition. Therefore, no problem here. No problem in the first generation. No problem here. Both of them are marrying somebody from outside. Therefore, they don't have the mutation. It goes generation after the generation, right, without any problem. Up to here, no problem. Both are heterozygous condition. But what happened after this? And now if you say that in a community, it can take any caste or tribe, two unrelated individuals, they don't know, they must have lost the root from where they have emerged. If two are unrelated, if they marry, obviously they are normal. There is a chance that mother contribute this abnormal chromosome, father also can contribute this abnormal chromosome, will become homozygous, and that cause the disease. So that condition is very high among Indian population or South Asians, right. So to understand, until then we don't have any clue whether those population which we included, 275 population, have any disease or not, because these samples have been collected for understanding their population history. Now we are predicting that there could be a population specific recessive disease, right. To understand that, we took 4 population from South India, we did the exome sequencing, and also we tried to understand by interviewing the population whether any disease in the population or not. In this 4 population, we found large number of deleterious mutation in at least 3 out of the 4 populations, right. So one of the population having high frequency of ankylosing spondylitis with high frequency of HLA allele. Another population we found a mutation which may cause cardiovascular disease in a high frequency. The third population, because the other 2 we are still characterizing, the third population is even more interesting, which has a disease called epidermolysis bullosa, junctional epidermolysis bullosa, which is a skin disease, and not only externally it has so much of skin lesions and so on, but internally also it affects, there is a lot of lesions and congenital heart disease also, where when we sequenced the exome, we found there is a 11 base pair deletion in a gene called LAMB3. Parents were absolutely fine, but they are carrying the mutation in a heterozygous condition. One of the pair is normal, other one is having the mutation, right. So initially when a novel mutation, we were not very sure whether this mutation is really responsible for the disease or not. So we wanted to screen large number of samples which we have in our lab, and also I mentioned earlier that recently we sequenced 10,000 individuals from 83 populations. We screened all of those. This was the one which the Honorable Minister of State for Science and Technology and some of us have released the data, and Honorable Prime Minister last year talked about that, and now this sequence have been deposited in Indian Biological Data Center in one of the DBT institutes. When we checked, none of these 10,000 individuals carrying this mutation. That means absolutely rare, right. We also looked at the global database consisting of 8,62,000 genome sequence. When we convert into chromosome double of that, 17 lakhs chromosomes doesn't have except in 2 individuals who are South Asian origin with heterozygous mutation, not homozygous. So that means so rare in a global scenario, but later on we found that it's highly prevalent in that particular population. Now it's very clear it's very rare mutation, but whether this mutation is really cause for the disease. When we looked at the protein, this is a full length protein 1172 amino acid. Because of the mutation 11 base pair deletion is truncated to 799 amino acid, right. So that's where the animal experiment would give us the valuable information. So using CRISPR-Cas, we knocked out that 11 base pair, and when we generated the animal, you can see that the homozygous one are not surviving beyond 2 days, whereas a human doesn't survive beyond 2 months. Those individual with the skin disease, they die within 2 months after the birth, where animals with the same 11 base pair deletion, they live only up to 2 days, but within the 2 days these scalings are happening on the skin, but we could not do much of the experiment on this animal. But when we looked at the heart and many other organs using microCT, I think you are going to get it soon in this institute. Dr. Gupta mentioned about that. This is a wild type and this is a heterozygous, where you don't see much difference between these two. Whereas homozygous, you can see the hypertrophy and also there is a septal defect. This could be the reason that why these pups are not surviving beyond 2 days, okay, they die. So what to do? Then we started looking at different clones with the different size of deletion. So one of that was 45 base pair deletion that consists of the 11 base pair also, where this is a non-frame shift, where earlier the 11 base pair deletion removes at least 380 amino acid, whereas this removes only 15 amino acid, therefore the impact is not very high. When we generated the animal model, up to 4 months there is no problem at all. After 4 months, slowly it's showing the symptoms as we have seen in the child, and 4.5 months what you see here, and beyond 6 months the animal doesn't survive, right. Before that itself it has all the abnormalities which we have seen in the patient. Now when we look at the internal organs, the heart is enlarged, spleen also enlarged, stomach become very small because the animal doesn't eat properly, and it has multiple other abnormalities, right. We are working on it, I am not showing most of the results. This is up to the animal. Now it is very clear this is a mutation which is responsible for the causing the disease and taking life. Now we don't have the human homozygous to understand what are the other biological significance of this mutation. Therefore we thought we generate iPSC from mother's amniotic fluid from the heterozygous. We made successfully homozygous iPSC from the human. Then we tested with, we transformed that into keratinocyte, this simple hematoxylin and humucin staining. We also used different keratinocyte marker, and this skin is heterozygous one is absolutely fine, whereas in the homozygous you can see a lot more abnormality, okay. So that explain in a human skin as the problem. We also transform into the cardiomyocytes, and you can see that we express LAMB3. In a heterozygous you can see the expression of that, but in homozygous very mild or completely absence of this. We also looked at the cardiomyocytes, and you can see how nicely it's beating in the heterozygous condition, whereas in the homozygous it hardly shows any beating, very, very mild, what you see here, and has been captured, you can see in the heterozygous, very nice, and homozygous it's not really beating. So all this suggests that some more experiments are going on, suggesting that yes, this mutation is real responsible for the disease. As I mentioned, we are looking at transcriptome, we are looking at proteome, we are also looking at the sperm and ovary and so on, but I am not presenting here. But now I am just shifting to human. What we understood so far, yes, this mutation is real responsible for the disease, then we have to do something for the families who have the disease. So went back, asked, is there any mother who lost children because of the mutation, is pregnant. Then we found a mother who lost couple of children, also pregnant again. Then we brought the mother from near Madurai to Chennai, collected the amniotic fluid and tested by simple PCR. This homozygous, heterozygous, and the amniotic fluid also shows the heterozygous. We further confirmed by Sanger sequencing. Then we advised the mother to continue with the pregnancy because the fetus carrying the heterozygous mutation, and about 2 years back the child was born with the normal phenotype and growing well, there is no problem at all. So this is the family who have lost couple of children, now they have, but the population level, if you look at, there are 5,000 individuals in that particular population. So then we thought that we should do something for the population, right? Then we approached Tamil Nadu government, there is a Directorate of Public Health and Preventive Medicine. After about a year long the paperwork, presentation and so on, they gave approval to go ahead with the collection of sample and screening in every single individuals. And with the strategy of looking at both every single individuals including children and also the pregnant mother, once we screen every single individual, blood or saliva sample, identify the mutation, and we wanted to create a database, somebody within the population can maintain that, and that would probably help in giving premarital counseling. Later, for example, children, school going children with the 10 years of age, after 10 or 15 years when they are ready to marry, take this sequence, also take the sequence of the individual whom he or she wanted to marry, see whether both of them are carrying the mutation, and we can advise accordingly. In the mother, we collect the amniotic fluid and again make the decision based on the genotype, and this way actually we can eliminate the mutation maybe after several rounds of screening. So that will give the population some kind of relief that without this disease they are in the future they don't have to really worry. So with this strategy, what we did, we went to that community. They are in extreme South Tamil Nadu. We had a camp, 2 days camp, 19th July last year and 20th July. We collected the samples and we tested and found 0.5% of the populations are carrying this mutation. You can imagine the worldwide it was not present at all, very, very ultra rare, but when you go to the population it is 4.5%, right. So having this, because now ultimately we are going to screen every single individual and give them the counseling. So knowing this thing for the last several years that there is a problem of rare genetic disease. So when I was a director of CDFD, we initiated a program called Mission Program on Pediatric Rare Genetic Disease. There are several organizations including um, some of the ICMR institute where Sudhakar also part of it when he was in NIRCH, and this is to screening the children with rare disease, and every single institute will screen that. If they don't find any mutation, then they send the sample to CDFD. CDFD will do either exome sequencing or whole genome sequencing, and if there is a novel mutation, as I mentioned earlier, they use different model system to understand the impact of that mutation. There is also treatment strategy for some of the selected disease with the help of CMC Vellore. We also have awareness program. There's a small video in 8 different languages so that the common people can go through that and get benefit out of that. It's a DBT funded program, 5-year program, and we established a laboratory in CDFD, and this was during the COVID time. Former Vice President of India, Sri Venkaiah Naidu, has inaugurated the lab, and later on Dr. Rajesh Gokhale, the Secretary DBT, has launched that particular program. It is going on very well, and any of you come across rare disease, you can refer them. They don't have to pay anything because this is a DBT funded program. So with this I want to conclude my talk by saying this, that whatever the population study which we started about 30 years ago, collecting samples, of course this is the Andamanis, collecting sample from the population for the population study. Now actually we are collecting the sample for understanding their public health. So these two are very well connected, at least from my own research. Later we are also looking at ancient sample which I have not presented. We are also looking at several other diseases which again I am not presenting here, mitochondrial disorders, various cardiovascular diseases including cardiomyopathy, male infertility, which Sudhakar has done his PhD, identified several novel genes, even infectious disease to understand the host genetic variation, how it makes susceptible or giving resistant against some of the infectious diseases. So with this I would like to thank all those who have contributed from my own lab, and there are a lot of collaboration, huge collaboration not possible. I think DG has highlighted about this collaboration. We do collaboration with so many institutes within India or outside India. And finally I want to thank the funding agencies, particularly DBT for Genome India project and the Pediatric Rare Genetic project, CSIR, UK India Education Research Initiative grant, DBT I already mentioned, ICMR. In fact, my first infertility funding came from ICMR long ago, that was about 20 years, 25 years ago. DST, Medical Research Council at London. So with that I thank, and if you have any question I will be happy to address. This also gives some kind of idea for the young colleagues who are here, that take a project, don't leave it in between, continue the project which I was mentioning. It's 30 years of work, right? Of course I presented only maybe 5 or 10% of the work which has some kind of connectivity, right? So take some lesson from here, and I am always available anytime you can contact for any kind of help, right? So thanks again, and if you have a question please.
For the wonderful talk. It's really I wanted to meet you. Thank you for this opportunity. Sir, actually I have one question. Before that I want to add that in Kerala, in the northern part of Kerala, Malabar area, some Mappila community is there. There they
Twins.
have Gaucher's disease. Gaucher's disorder.
Okay.
So in that GBA mutation is there, and they have non-neuropathic disease is there.
Okay.
So I think I have seen your one slide that you have taken samples from a different part of India, southern part, but not from Kerala. So
Okay.
please try to collect from that also, and please do some.
No, Kerala also we have samples.
Yes, sir.
Recently we studied
Yes, sir.
coastal region of
Yes, sir.
Kerala and in fact Karnataka,
Yes.
Kerala also. But this particular community we did not look at it.
Yes. Sir, I have one question because earlier I did a complete mitochondrial genome analysis of Indian cattle for the first time, and we observed a different, so many salient features. So I am not discussing about that, but I have one doubt. So if we are collecting some fossils, so can we own the fossil? It's otherwise it is a government, or can we own as a public? We can own that fossil, and if you want to some genetic analysis of that fossil, we want to get any permission from the government or Geological Survey of India and all. Otherwise, can
So,
we do without that permission?
Which fossil you got?
For example, for example, 3,000 years ago, we are getting some fossil of some cattle or buffalo or something.
Now, if you are excavating,
Yes, sir.
and you can do whatever you want to do with
So
that.
I want to study any genetic
you
analysis.
can-
You want to take any permission from the government?
No,
or?
not necessarily.
Okay.
If there is a sample in either Archaeological Survey of India or Anthropological Survey of India, they have their own museum. You wanted to collect from them, of course you have to write to them and give it.
I'm going to the field. From that I am collecting.
Yes, you can collect, but only problem with the fossils are you may not get DNA from there.
Yes.
So unless otherwise DNA you have, you cannot do any kind of
So
work.
that is, I have seen that Kayajin, they have ancient DNA isolation kit and all. So
Yeah.
I want
So-
to try, but I want to ask you because I want to see if I'm someone person to ask the question. So that's why
You can try, but you need to have a special ancient DNA laboratory. Otherwise, what happens? You will contaminate with your own DNA or any other, if it is like a cattle or some
Our
other
own DNA,
animal,
yes.
but there will be a contamination from different sources. To avoid that, the ancient DNA laboratory is very typical, not very big, small. There are 3 rooms, positive pressure always, and U is always on, and you have to be very careful in handling that, otherwise you contaminate. So what happened, because in the ancient samples, they're all very fragmented, right? But when you do a PCR, even few molecules of fresh DNA can amplify, take over. It will not amplify the old ones, right? So you have to make sure 100% there is no external contamination. You are trying to amplify only the ancient samples.
Thank you, sir. Thank you very much.
Okay. Thank you. Once again, wish all of you very best on this particular activity.
For sharing the research journey with all of us. So now I request our director, sir, to kindly felicitate Dr. Kumar Swami Thangraj, sir. This is a token of our deep respect and appreciation for all the efforts you have done for the science. Thank you, sir. Now we move forward for another enriching today's oration by our guest of honor, Dr. Naveen Kumar, sir. Sir is the director of, we all know, ICMR, National Institute of Virology, Pune. He has done his postdoctoral research at the School of Medicine, Emory University, Atlanta, USA. He was a visiting fellow at Edinburgh Medical School, University of Edinburgh, UK. He has served as a head of the National Centre for Veterinary Type Cultures at Hisar. He has made significant contributions to understand the virus-host interactions. Dr. Kumar is the first to isolate the lumpy skin disease virus in India and developed the world's first DIVA or LSD. He is the recipient of several awards, such as Award of Excellence in Agriculture Research, India Animal Health Award, the Commonwealth Professional Fellowship, Societal Innovation Award from the Ministry of Science and Technology, DART Fellowship. He has served as a technical advisor to WHO and WOAH on several infectious diseases. He is the section editor for the General Virulence and has been a reviewer for several international journals. Thank you, sir. The stage is yours.
Chief guest of today's function, Dr. Thangraj, Director Dr. Gupta, all the distinguished scientists, technical staff, students, ladies and gentlemen. So I congratulate all of you for this 4th birthday of this esteemed institute. So don't take it lightly, you know. अभी सोचिए कि राजस्थान रॉयल में if there is वैभव सूर्यवंशी, you know, out, so this it seems that now they are going to lose the match. So you are 4-year-old and NIV may be 75-year-old, but we are highly dependent. Within 4 years, you know, NIV is highly dependent on this institute. So this is Vaibhav Suryavanshi of the ICMR. So, as our Director General already told, and Dr. Thangraj has given a very beautiful lecture, you know. So, whatever the research that is been undertaken, so is less than 1% is being translated. And one of the limiting factor, particularly in the biomedical research, is because of regulatory issues. So, even it applies to everybody, you know, whatever the research I have done, the paper I have published. So if you look back, you know, hardly very few, less than 1% resulted into the development of any product or technology. So this institute is actually not going to serve a key player in the biomedical research within ICMR, but also among other research organizations, including DBT, ICR, CSIR, DRDO. So because this is a unique facility, this is not available with any other organization. So that's why I'm again saying that this is going to be a key player in upcoming days. Regarding NIV, so two of the important projects, one on KFD. So the NIV can do the laboratory research, characterizing the virus, making the vaccines and all these things. But eventually if you want to take the product to the end user, you know, so then you have to undergo the preclinical, you have to produce these data as per the regulatory requirement. So the facility is only, you know, with us in ICMR. So that's being, I also, you know, feel proud to be part of the organization that we have such a facility in the ICMR. So besides KFD, that we already actually mentioned by our Honorable Director General, sir. So there is one disease, you know, maybe many of you don't know, Chandipura. So Chandipura disease is actually, it was, the virus was invented in 1965 by NIV. And since then, you know, disease comes after, every 10, 12, 15 years. So major outbreak took place in 2003, where is more than 300 children died. So Chandipura is a virus very much of the rabies family. And then, a lot of work is undertaken, but still many of the things we don't know. 2 years ago, there was an outbreak in the Gujarat where about 60 children they died. So it infects the kids, children's, and the sudden after infection there is fever, and next morning with 24 to 48 hours after the fever the children they die. So many of these aspects we don't know, and that's why we have initiated the research project to understand how about the pathogenesis of this virus, how virus travels, because there is sudden death. So if you want to take the samples, maybe blood or CSF sample and try to test, many times we miss the virus, because what is the appropriate time we don't know, where we should look for the virus. So in the first few experiments that has been successful, this morning I was talking to the director, the outcome of the experimental infection in the guinea pig has been remarkable. So we don't think we need to go beyond looking for other animals. Initially it was planned that we will test multiple small animal and large animal, because if you want to test the vaccines or the therapeutics, so you must have a small animal and then a large animal model before actually going to the humans. So these are the two projects, and other projects that are actually we are likely to initiate. So I am again going to contact you. So your importance is immense, and one thing I actually want to convey to the scientists, so that you think that why this institute cannot be a best in the relevant field. So what are the limitations? Why you cannot be a best scientist? So if you ask this question, so then, so do we are lacking the infrastructure? Definitely not. So 25 years ago, I first time went to Germany for my PhD. So at that time, there were very few institutes in India. And I was, surprised to see their labs and everything. But today, each and every ICMR institute is world-class infrastructure. So we don't have, problem of the infrastructure. So second thing is that, do we are lacking in funds? Definitely not. So funds, it's very difficult for me and for the director as well. So हमें डीजी साहब की डांट खानी पड़ती है, तुमने खर्च नहीं किया. पैसे की कोई कमी नहीं है. Infrastructure is beautiful, world class. See, the only limitation is that is our thinking. So science never depends entirely on the technology or the resources. It depends on how people think. So we have to think big, Why you can't be equal to the researcher who is working in the Harvard or in the Cambridge or any good institute across the world. So we have to change just our thinking so that we can actually do the wonders. So once you are having your PhD degree, so you are independent researcher. So you have to think that you can independently think and execute your work. So this year, among all the research organizations, so ICMR is the only organization where we have an increase in 27% of the budget. Other research organization like DBT, CSIR, they are marginal, less than 1%, and one of the organizations even decreased. So you must feel proud that you are part of such a great organization, where there is a world-class infrastructure, where you have sufficient money and all other resources that are required. So only thing is that we need to think independently, think big, and execute to lead this institute and also to the ICMR at any best institute or organization in the country. So I again thanks to the director all the scientific staffs for inviting me. I must not be considered as a guest because I plan to visit here to discuss the outcome of the experimental infection, but this was one way that I was formally invited. So thank you very much, and I wish you again a very happy birthday. Thank you.
Thank you so much, sir. On behalf of all the scientists, we all will try our level best to keep the hopes of all seniors. Now, I would request our director, sir, to kindly felicitate our guest of honor as a token of respect and appreciation. We are truly honored by your presence, sir. So now we all will take here a short break and will continue with some of the sports events which are still incomplete. And in evening, we will meet again. Thank you all.